All right, girls. So where I left off was talking about interphase of meiosis and how synthesis has to occur before the cells can start to divide. So we have to make a copy of our DNA. So that's going to happen in S phase. But the other thing that's going to happen by the end of interphase, besides the chromosomes duplicating, is that the centrosome is also going to be duplicated by the end. And so somebody was asking me, what's the difference between the centrosome and the centrioles? The centrosome is the location where the centrioles are found. It's just a part of the cell where the centrioles are always found. And it's going to duplicate during interphase because you need to end up with another pair of centrioles on the other side, because remember, the centrioles are where the spindle fibers come from, and the spindle fibers are going to be what actually connects to either side of the sister chromatids and pulls them apart onto opposite sides of the cell during anaphase. So you have to make sure they have a copy of that too, along with the other organelles and things that are going to be necessary. So by the end of interphase, the chromosomes are still going to look like chromatin, because remember, they don't condense into chromosomes until prophase. So let's move on without further ado. So the first step of meiosis 1, or the first meiotic division, is going to be prophase 1. It's the same order as mitosis, except it's going to happen twice. So prophase 1, the chromatin is going to coil up into the individual chromosomes that you can actually see with a microscope if you were to take the cell and put it under a microscope with some stain. In a process called synapsis, homologous chromosomes are going to come together as pairs. So that's what you're seeing over here. You have this homologous pair come together, you have this homologous pair come together, and this homologous pair come together. And that's a process called synapsis, is when they join up and find each other inside the cell. And that's going to happen in prophase 1. And then the resulting group is called a tetrad. Tetra is 4, so tetrad. So there's 1, 2, 3, 4 chromatids as part of this tetrad. From there, something really unique that happens in prophase 1 and only prophase 1. It never ever happens ever again. And we actually don't even know why it happens at this point in the first place. We know that it results in genetic diversity, but we don't know why it happens at this point, and we don't know what's controlling it. So scientists are doing experiments, probably as we speak, trying to figure out why or what signal is telling the cell in this phase to undergo this process that we call crossing over. So if you look, you'll see that at some points, the tetrad, or the homologous chromosomes, are going to like cross over on each other like this. Everywhere I've drawn this little red dot. Okay, that's a spot where they cross over. Well, a phenomenon that is called crossing over, because here's one thing we can give an easy name to, basically parts of the chromosomes are basically just going to switch places. Remember, these chromosomes are homologous, so it's okay for them to cross over because the same genes are there. It's just different versions of the genes. And so they're going to basically just switch places, which is going to happen in prophase 1, but in this picture, it doesn't really show you what that looks like until this anaphase 1 picture over here. But anywhere you see that there's like a blue and a red piece, that's a place where crossing over has happened. So a piece of mom's chromosome will go and attach to a piece of dad's chromosome, but it only happens in the homologous pairs where the genes are the same. So the genetic information is not being mutated at this point. People always think that this is a mutation. It's not a mutation. It's just a point where the chromosomes, if they are paired up with their homologous partner that has the same genes, but perhaps different versions of that gene or different alleles, hopefully that term is becoming familiar to you, allele, but the alleles can switch. So let's say that this blue one right here, right, let's say that the gene for eye color is found here. And the blue one says that this baby is going to have blue eyes, but the red one says that the baby is going to have brown eyes. Well, in this case, the brown and blue are going to end up switching places. So even though the rest of the chromosome is exactly the same as it was, it's not going to be changed in any way, you now have a piece of what you got from your dad found on the mom's chromosome, and a piece that you got from your mom found on dad's. And I'll see if I can find another video that kind of simply only explains crossing over and shows it with animation. But it's this really amazing phenomenon that's going to allow for a lot of genetic diversity. Because if you notice over here in anaphase, right, this sister chromatid had crossing over occur, but its partner, the other sister chromatid that was genetically identical, is now genetically different. So that's how in the very end, when we have those 4 daughter cells, that's how we really end up with them being genetically different. There's a couple of other things that we'll talk about that go into their genetic differences, but crossing over is a really big part of what causes them to be genetically different. And again, scientists don't know what's controlling this, and it's completely random. Sometimes chromosomes will cross over, and sometimes they won't. Literally completely random. Coolest phenomenon. I love to think about it. I love to talk about it. If we were in class, I know you guys would have tons of questions about it, so I'm doing the best I can in the screencast and with like the tone of my voice to express to you how cool this is. It really allows cells or gametes to be so genetically diverse. One egg cell from another, one sperm cell from another. Super duper cool. But only happens in prophase 1 of meiosis 1. Never happens again. So yeah, that's prophase 1. So, crossing over, like I said, I didn't realize I had a slide on this, haha. It's vital for creating genetic diversity, like I was just saying. Basically, the chromosomes are just allowing themselves to change, but not mutate. It's not mutating because the sequences are not changing, it's just the version of the allele that you're switching over is what's changing, that you're crossing over. But anyway, by the end of prophase 1, the chromosomes have condensed, the centrosomes are starting to move to opposite sides of the cell, so that's what you can see here, they're starting to move to opposite sides, and the spindle fibers are starting to form in between them. And then the last hallmark of prophase 1 is that the nuclear envelope, all these little purple pieces, the nuclear envelope starts to disappear. Because remember, we don't want a nucleus when we're trying to pull chromosomes apart. The nucleus would get in the way. And so the tetrads are going to start to move towards the center of the cell. That way, by the beginning of metaphase 1, the tetrads are now lined up on the metaphase plate, or the equator of the cell. Okay, so they are lined up on the metaphase plate, and it's not going to be until anaphase that the tetrads actually get separated from each other. Crossing over has already happened by the time we are in metaphase 1, and the spindle fibers, by the end of metaphase, the spindle fibers are either attached to the kinetochores, that are little proteins found at the centromere, which is this little spot. Well, let me write. Yeah, there we go. Which is this little spot that's holding them together. So the microtubules are either attached to the kinetochores, or they are pushing against each other. So the spindle fibers, not only do they grab onto the chromosomes and help pull them apart, but they also push against each other in order to help elongate the cell. But yeah, so the spindle fibers are only going to attach onto the kinetochores of one of the parts of the tetrad. And so that way when the cell starts to divide, like during anaphase, which is like the next picture that's kind of shown on this slide, that way the tetrads are being pulled apart. So we're not pulling apart the sister chromatids from each other yet. We're just pulling apart the homologous chromosomes. That is the major hallmark of metaphase 1. And I do want you to be able to recognize a picture of this versus a picture of mitosis metaphase. If this was mitosis, instead of being lined up side by side like this, they'd be lined up in a straight line. So anaphase 1, the spindle fibers have started to pull the homologous chromosomes apart. Sister chromatids are still attached, only the tetrads are moving. That way, in this picture, you still see that they're attached to each other at the centromeres, or at the kinetochores, if you will. And then in telophase 1, you start to get the development of the cleavage furrow, which is just the little, I guess, crease that's forming between the two cells. Basically, at this point, the chromosomes of the homologous pair have each arrived in their own end of the cell. Cleavage furrow is almost completely formed, and then cytokinesis will happen right after in order to fully separate these two cells. And by the time telophase 1 and cytokinesis is over, we will then start in meiosis 2. So this video is only 12 minutes, but I'm going to stop it here. That way I can talk about all of meiosis 2 in one fell swoop.